right? So this would be the origin or the top, right? That's where the wells are, and this is the bottom. And remember when we were talking about a sequencing reaction, we had, if I just drew lines, because that's easier. Look, there's double-stranded DNA, yes. And here's our sequencing primer. And of course, the 3' end needs to be on the correct side, so it's going this way, right? And so it would be complementary to this strand. And Taq DNA polymerase is going to complementary base pair to that strand. Remember, we added in those di-deoxyribonucleotides with, what are they missing on the end? Yeah, they're missing the juicy 3'OH. So there's just an H there. So that chain terminates, right? And that just means DNA polymerase can't grab that OH to add the 5' phosphate to make the phosphodiester bond. Right? So then the DNA polymerase falls off, and however far that one got is as big as it is. Right? And so, right, if we're in, and each lane, this has all the normal nucleotides plus the A chain terminator, the missing oxygen on a T, on the C, and on the G. Right? So in this row, all the fragments end in A with that dideoxy guide. And likewise, T, C, and G. And remember, the small fragments run to the bottom of any kind of electrophoresis gel, right? Just like the ones we did for restriction, digest, and PCR. That's just a little bit different type of gel. This one resolves down to 1 base pair, right? Whereas the other ones, I said you can tell the difference between 900 base pairs and 1,000, but you can't tell much more than that on those other kind of gels we were talking about. But it's the same idea. We're putting the DNA mixture in the wells. We're running electricity. DNA is negatively charged, so it runs to the positive pole. The smaller fragments go faster. And so wherever it stops, we'll get a piece that size that ends in an A, wherever the next A is, and in this reaction, there's only 2, okay, and so on. So when you're reading the sequencing, this would be the first base pair. That's the smallest fragment, right? We generally can't see the sequence of the primer. We're going to, when we actually do these reactions, right, that's what the small little pieces being sent through. The first one that's fluorescently labeled is part of the newly complementary. The primer's not labeled, right? So if it's just primer alone that goes through the gel and into the machine, it doesn't fluoresce, so it doesn't show up as anything, right? So the smallest piece would be the primer plus the very first base pair, which in this case would be what? T, right? So this would be the primer. Oh, look at how cute he is. With a T sitting there, right? So that would be this guy. So that's a T. Let me get us another color, and that's easier. And then the next size fragment is this one. What's really going through the machine is this plus the T and an A, right? Because this is in the A lane. Yes? Yeah? Right? Because it chain terminated at A, right? It just added a normal T, but then, that bad A got stuck in there, so it stopped. Right? It was Taq DNA polymerase was grabbing on, right? This was the original 3'OH of the primer. Primer's in red. And then the next size up, right, is this guy. He's in the C lane. We'll add him in here so we don't forget about little A right there. Okay. And so that piece is really, here's the primer, T, A, C. Right? C is the one without the oxygen a chain terminates at that one. Okay, and so you just continue to read up. There's another T, then this guy's a G, that's the A, T, I'm getting too far, and C. Okay. And so if you're asking the question, well, which one's on the 5' end, right, these are the primers. So then this has to be the 3', this has to be the 3' end. That's where it's adding to. So the most 5' sequence is that very first one. So this is the 5' end of your sequence. This is the 3' end of your sequence. Okay. So if you were asked to write it out left to right, 5' to 3', right, this is how we'd read the sequence, blah, blah, you know, dot, 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 because I can't fit it, like that. So on a multiple choice question, if this isn't a written question, you can't just write it along the side. You'll be looking for something left to right. And so you want to make sure you know that this going this way is that going that way. Yes?
But isn't the sequence of the genes supposed to be complementary to that?
So the sequence that you get out of the machine is this sequence. Yeah, and then depending on which strand you stuck the primer on, right, this tells you if you're talking about the coding region or the non-coding region of the gene. Right, but the sequence of the gene is double-stranded. So if you wrote out this and put it double-strand, that would be correct, right? But yeah, so technically the sequence you get out is complementary to whatever you're priming off of. So yeah, if you're priming off of the sense strand, right, then you're going to get the antisense or the non-template, and then, yeah, reverse and complement or the complementary strand is your template strand. So yeah, I'm not going to be that picky. Okay, so if I'm asking for a sequence of the gene or whatever, I know there was one homework problem that wanted you to do just that. This is the sequence you got off, but your primer, and they went, your primer sat on whichever strand, and then they made you, but I won't make you guys do that for an exam. If you understand, you should be able to understand what they mean by that, because, right, the gene is really double-stranded, but usually we talk about the sequence of a gene as the sense strand, right, which, complementary to the, right, so exactly. But if we made the primer going the other way, then the sequence coming off the machine would be the sense strand. So it just depends on where we put the primer, right? And usually when we're sequencing, just to make sure we're correct, we'll sequence go through the gene in both directions, so that if there were any mistakes in this process, either that the Taq polymerase made, or just the machine read wrong, or there was a fluorescence that went crazy, you could double check, right? Because yeah, if you sequence going this way, and then you sequence going that way, those should be complementary. So if you then reverse and complement one, you get the same sequence. And if there is a mistake, then you can resequence and see which side was correct. Exactly. But for you guys, I'm just going to tell you, whatever's on this gel, I want you to be able to read a sequence from that. Okay? Is that good? Is that clear? So it's not confusing? Okay. So screenshot this and upload it. So write out your sequence, take your notes, whatever we normally do. You should be doing this along with me, because I like that. Makes me happy. It really does. It's really sad, but I love seeing people taking notes and asking me questions. Do sequence practice, should be the one between the two. And then you guys are going to do this one as the sequence after. Oh, yeah. Sorry. So if you've screenshotted and uploaded this, you can go ahead and work on the next one. Yeah. Uh-huh. Yeah, I just ran out of room when I was writing it out. Yeah. Yeah, I was just like, in a multiple choice, they'll be written left to right, not along the gel. So I want to make sure people realize reading it up is essentially reading left to right. But then I got scribbly and missed, and I couldn't fit it. You always read it up because those bottom pieces are the ones that come right off the primer, so you know where you're starting from, right? Your primer, you know the sequence of your primer, and so that means you know where it sits, and so then you can find it. If you start somewhere up at the top, you don't know. I mean, you could read it down, but then it's essentially 3 to 5, and you'd have to switch it around anyways. So yeah, the smallest ones, and in a real gel, a lot of times that the smallest piece you can see on the gel isn't always that very first base pair after the primer, but it's the closest one that's resolvable to the primer. So you always know where to orient yourself, starting at the bottom. Start small and then get bigger and bigger. Yeah. Just keep on writing. It's probably that little ghost that just didn't come out very well. Yeah. And sometimes that would be like, if there's a blank, you're like, I don't know what that is. So then you look at the other strand and go, oh, that was just an error pipette, something crazy happened to that little one.
That, when you're reading it, for example, the primer is going to have like T, for example, and it terminates because it doesn't have any more. It doesn't have anymore.
that was the fluorescent T.
Fluorescent. Okay.
Yeah. Or radioactive, but it had the missing O, so DNA the polymerase can't add any farther. So wherever that T got stuck, it just stopped. And since there are some normal T's in that mix also, it doesn't always incorporate a bad T, the first T. It could incorporate a normal T, the first T, a bad T, the second T that comes up, so then you get a bigger piece, or the third T. So randomly, in this reaction, there'll be pieces of all the different places where there's a T. Normal, 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 normal. Oh, bad one. Oh, a bad one was first. Ooh, the bad one was third. Ooh, so you'll get every single one represented at least once in this reaction.
So the bad one is the fluorescent one?
Yeah, the bad one is the one that's fluorescent and that's missing the O. You can't make a phosphodiester bond off of it. Yeah. So from the bottom's always the 5' end, because that's the smallest piece, and that's the one coming off the primer, right? And so the end of that is 3', because you add the 3' end, so the beginning is the 5'. Yes? Okay. Yep, so then it's just that reverse transcription, and then regular PCR. Yeah, so the RT part goes RNA to DNA, second strand, and then you're just doing PCR just like you normally would with those 2 primers.
Okay, cool.
Yeah.
How would the primer show up RNA? If we're doing DNA, how come the RNA shows up?
How come the RNA shows up?
Yeah, like the RNA primer, if that's closer to the primer, how come we can deduce that's the primer if what we're doing is doing the DNA is
So that primer is DNA.
Oh, it's called the RNA primer?
No, we only in real cells during replication is the primer real RNA. When we're doing anything in a test tube, we use DNA nucleotides because they're more stable. But we could use RNA, and it wouldn't matter, right? Because that 3'OH is still exactly the same. The polymerase doesn't really know the difference, right? And it shows up because if it was naked by itself without a fluorescent label or without anything, then we wouldn't see it whether it's DNA or RNA. So it has to have one of the nucleotides that are fluorescently labeled, and the only ones fluorescently labeled are the chain terminating missing that O. Right? So it's always the primer plus nucleotide. If it's in the A lane, that last one was the bad guy that stopped it. Right. So you can't get any farther. Polymerase falls off. It can't. I'm trying to put. I'm done. Right? Yeah. Yes. Yeah, and that's close enough. Yeah. So you get most of it. Yeah, and that's the other thing, like, when you get too long of a run, it's real hard to read, and so then you'll make, in the real world, you'd make another primer close there that overlaps where you can still see to get farther, and then overlap to get, so you put together all these sets. And now we can get, the fluorescent kind is, you can get like 500 base pairs per shot, so much easier than the gel. All right, screenshot, upload the sequencing after for that, the, whatever I'm talking about, whatever we did, this one. After you did this guy, just get up as far, you don't have to do these all the way up to the top because they get all fuzzy and close. All right, what else are we doing? I have scribbles on this. Did we talk about this? Yes? Oh yeah, so this was just the same kind of sequencing reactions except with the fluorescent label. The ones in the gel really are radioactively labeled. It's the same exact idea, right, where the chain terminator is missing that oxygen, it's fluorescently labeled. That gets run through a gel by size, and it's actually sent through a machine to read the fluorescence, and then it actually gives you the sequence so that you don't even have to read up the gel. So I won't give you that one, because then I'll say, give me the sequence, and it's already written. Or should I? Would that be good? Okay. Can you guys get that? Yeah.